Welcome everyone. So today we will be discussing about the next separation technique, which is chromatography. Now if you will just look at the heading of this topic, then it is not given there, but this topic, it is all based on chromatography technique. Now how this technique or how this method is done, that is what we will be discussing in this activity 2.7. So take a thin strip of filter paper, draw a line on it using a pencil, approximately 3 centimeter above the lower edge. It is also given in figure 2.8. So definitely to understand this activity more clearly, you can refer to figure 2.8a. Put a small drop of ink, water soluble, that is from a sketch pen or fountain pen at the center of the line. Let it dry. Lower the filter paper into a jar, glass, beaker, test tube containing water so that the drop of ink on the paper is just above the water level as shown in figure 2.8b, and leave it undisturbed. Watch carefully as the water rises up on the filter paper. Record your observations. So this is what is given in activity 2.7. Now first of all, to understand this activity, let us just refer to the figure given for this activity. As you can see that figure 2.8, it is showing here the separation of dyes in black ink using chromatography. And this figure, it is also having two parts or two sections, A and B. So A is actually showing that what are the things we can do wrong, whereas B is just representing the completely all the right informations which we need to apply to perform this technique. Now before this, I just want to make you understand that what kind of mixture we have to take. That means the nature of mixture which we have to take, so that on what type of mixture we can apply this technique. So if you remember the first technique which we discussed, the first separation technique which we discussed, it was evaporation. And evaporation technique we applied for what kind of mixture? For that mixture in which one of the component is volatile and the other component is non-volatile. And for that technique we took the mixture which we taken, it was ink. So that ink, you can take any ink like ink of the fountain pen or any ink. So that ink we have taken. Now ink, it is a mixture. It is a mixture of water and dye. Now water you all know. Now what is dye? Dye, the general term or the most commonly described term for dye is it is a colored component. Suppose if you are just looking at a dye and it appears to you blue or any other color like green, it can be of any color for sure. So blue, green, black, red, any of the dye which you are taking, it is not a single color or it is not a single component. The dye which you have got in that separation technique of evaporation as a residue, that again it is not a single component, it is not a single color, it is formed by 2 to 3 different colors to get the desired color of what we want. So that is what, how to understand that. Suppose if you are having a dye, then how to understand that this dye it is made up of 2 different colors or 3 different colors or 4 different colors. So to understand and to know that how many components and what are the components, what are the colored components are mixed to make this desired dye, for that we are performing the technique of chromatography. So there is a difference between the technique of evaporation and chromatography. For evaporation, we have taken ink. Here also we are taking ink, but there we are having different aim. We want to know whether the ink is a single component or not, and we found that ink is not a single component. Ink is a mixture of water and dye. And chromatography technique we are applying to know that dye, is it a single component, or dye again is a mixture of 2 or 3 different color components. I hope that you understand what I am saying, because this is where the students are getting confused. So chromatography technique we are particularly applying to know that whether a dye which is a colored component is again a single substance, or it is again a mixture of, it is a mixture of 2 or 3 different colored components. Now this is very important, very important point. And to know that, we will be applying this technique of chromatography. So here what you have to do, you have to take a filter paper. Now for chromatography, see, there are many different type of filter papers are available. Generally we are using filter paper for the process of filtration. For chromatography technique also, the paper which we are using, that is also filter paper, but that is particularly it is a somewhat different from the other, that means which is used for filtration. For chromatography, the filter paper is particularly specialized or for the process of chromatography only. So there is somewhat different in nature and in texture. So here what you have to do, you have to take a filter paper as it is shown. Just let us first discuss about the A part of figure 2.A. So the filter paper you have to take, and from the lower edge, as you can see, from the lower edge you have to measure 3 centimeter above, and you have to draw a line with the help of pencil. Now again it is very important, it is a point of precaution also. When you have to draw a line on a filter paper, then you have to draw it only by pencil. You can't use a pen, you can't use a sketch pen or any other thing which is having ink. You have to use a pencil, because pencil is not a ink. There you can't use a ink, because here the result which we want to know, for that we are using ink. So after just drawing the line above 3 centimeter with the help of pencil, then in the middle of the line which we have drawn, there we have to place a very small drop of ink. That is what it is given here, spot of ink. Now you just see that a spot of ink here where they have taken in the A figure or A part of figure 2.A below the line which is drawn. So that is wrong, and that is what it is showing. Now in B part you just see that the spot of ink, it is exactly on the middle of the line which we have drawn, and it is also not above or below the line. So this is important, and that is why now we will be discussing about figure 2.8b only. After that, what you have to do, this filter paper you have to suspend with the help of a glass rod, with the help of a pencil or a ruler, in a beaker or in a jar or in a test tube, whatever the things are available in laboratory where you are performing. So just you have to suspend this, and that jar, here they have taken jar, that jar should be filled with water. Now the level of the water should be such that it should not be touching, or it should not be above the line, it should be below the line, but the filter paper, the part of filter paper, it should be dipped. So you can see that the lower end of filter paper is dipped in water, which is around 1 or 1.5 centimeter. That is the part of the filter paper dipped in water, but we have drawn the line above 3 centimeter from the lower edge. So the line should be above the water level. So this is also a point of precaution which is very important. Then like if you are simply at home also you can do, like if you have taken a cotton cloth or a paper, and simply just a lower end also, a small part of that cloth or paper, if you are dipping in water, you can see that water will eventually rise. It will rise slowly, but definitely it will rise. So the same thing we are also going to observe here. Here also water will rise above the filter paper. Now, if you in the activity, one other very important point is given, which is that ink is soluble in water. The ink which we have taken for this activity should be soluble in water. So when water will start reaching towards the line, then there at the line we have placed a spot of ink. That ink is soluble in water, so that ink will dissolve in water, because water has already reached there. So water which is present in filter paper, ink will dissolve in water, and when water will rise above the line, above the spot of ink, now ink will also travel with water towards the upper part of the filter paper. And when ink will also just ink will also travel to the upper part with the help of water, as it is dissolved in water, then you may find that you are either getting 2 or 3 different spots. So that 2 or 3 different spots at the different heights on the filter paper, it clearly shows that dye, it is not a single component. It is also having, if you have got 2 spots, 2 different spots, then you will be able to find that the dye which you have taken, the dye which was present in ink, it is having 2 colored components. Or if you have got 3 spots on the upper part of the filter paper, then it will imply that it shows that dye which is present in the ink, it is mixed, it is a mixture of 3 different colored components. So with the help of this chromatography technique, we will be easily able to understand that whether the dye is a mixture of 2 colored components or 3 different colored components. So that is what you will be able to observe. And just to make you understand about the chromatography technique more clearly, more properly, I also shared one one video with you all. So if you have watched that video, it will be much easier for you to understand, because that video, it is they have shown or they have represented this chromatography technique very nicely, very properly, and how the colored components of the dye, they are also getting separated at different heights, and what is the reason that they are, or you are able to find the colored components at different heights. So this is very important. Just go through the previous video, you will definitely understand. So this is about the chromatography technique. I hope that you have understood. Only this much is there. It is very easy to use. It is not even time consuming also. Hardly within an half an hour, within 30 minutes, you will be easily able to get the result. And on the basis of this, we will be discussing about the question and answer, which is just given below this figure, as you can see. So that we will be discussing in next class. So just if you have watched this video, if you haven't watched the previous one, just go through the previous video. It will be easier for you to understand, as because of the different colors, and they have shown it very nicely. Thank you.